All right. Good morning. Okay, so I think we stopped here, right, last time, talking about the change of fuel utilization during the exercise from low to moderate to high intensity exercise. Remember, we talked about the two, different energy fuels, carbohydrate and fat utilization change during the whole time period of exercise, depend on the intensity of exercise, right? And during the whole period of time from low to moderate and high intensity of exercise, our body is able to constantly use the carbohydrate oxidation, right? So carbohydrate oxidation, we pretty much, increased consistently, but the fat utilization is different, right? From low to moderate exercise, the fuel utilization is increased, but start from moderate to high intensity. As the intensity increase during exercise, our body utilize fat. The ability to utilize the fat has actually decreased. Remember this, right? So that's why we emphasize the carbohydrate store, particularly muscle glycogen store, is very important for the exercise, the efficiency, and because of, again, during the late stage from moderate to high intensity exercise, the fat utilization decreased. So that means that in this particular time period, our body or particular muscle is heavily reliant on the carbohydrate utilization, right? So if you have a more carbohydrate stored in the muscle, then you'll be able to utilize the carbohydrate to produce ATPs and to allow you to continue to perform well. But if you have a lower carbohydrate store at this stage, then you have less ATP being produced during this period, right? That will significantly reduce the efficiency of exercise. So here I wanted to just give like an example of a healthy young male exercise the subjects during the 2 hours exercise and the 1 hour post-exercise recovery period, and see how energy fuels in the circulation changes. That will tell us, give us some idea and what type of energy fuels available and used for the ATP production. So for example, during the 2 hours of a exercise period, you can see the glucose level actually declines, right? So glucose levels decrease. The lactate levels increase, which makes sense, right? We have utilized a lot of carbohydrate during this time period and produce a lot of lactate, not necessarily from skeletal muscle, but from other tissues, right? Remember, during the exercise, muscle becomes the most important tissue that we need to save the glucose for muscle use the energy, right, during exercise. So the other tissue may just go to the partial oxidation of glucose, so a lot of more glucose being utilized by skeletal muscle for energy during the exercise. So you see the lactate production is increased, the secretion from the other tissues that indicate the glycolysis, particularly the partial oxidation of carbohydrate in the other tissue may increase, right? And if you look at the fatty acids is also important fuel, right, for muscle in the exercise. You see the also gradual increase the fatty acid levels in circulation. That means the fat tissue may break down triglyceride and release fatty acids into circulation, and this process called lipolysis, right, increase during the exercise. And we're going to explain how fatty acids release or lipolysis is increased during the exercise. That actually regulated by hormone levels, right? And glycerol levels is also increased, a similar pattern, right, to the fatty acids levels during the exercise. It's also indicated the lipolysis, right? But lipolysis, triglyceride breakdown release both fatty acids and glycerol. Yeah, and triglyceride level is not much change, right, during the exercise in the circulation. And during recovery period, you see the glucose levels is going up again, right? Return to the levels, before exercise, and fatty acids going up a little bit more, but then also decrease, right, during the recovery period. And lactate level is significant dropped, right, and glycerol level is also decreased. So that just give us some idea during exercise, because of muscle utilize a lot of glucose, the glucose levels dropped, and the fatty acids levels increase, right? So muscle takes up a lot of fatty acids and use as energy for ATP production as well. And then look at the hormone levels change. The same subject, and 2 hours exercise and 1 hour recovery. You see that insulin levels significantly decrease. Why insulin levels decrease during the exercise? Say that again. What factors that stimulate insulin release, remember? Glucose, right? So insulin decrease here is because of glucose level decrease, right? We just talked about the last slide. So insulin levels decrease because of glucose level drop, and glucagon levels increase with same reason, right? The decreased glucose levels leads to increased glucagon and decreased insulin levels. And you see the catecholamines, norepinephrine, epinephrine levels will also significantly increase during exercise. This is very important information, right? The catecholamine are released during the exercise. So remember, the catecholamine is a very important hormone that stimulates lipolysis, right? And insulin basically suppresses lipolysis. So this patterns of the hormone levels, like increase catecholamines and decrease insulin levels, and will lead to increased lipolysis, increased fatty acids release, right? So in other words, that the reason why lipolysis increased, fatty acids release increase, is because of hormone levels change, right? Decreased insulin levels, increased catecholamine are the main driver, for the fatty acids released from adipose tissue. Now let's talk about the physiological change during exercise. We actually basically already talked about it in the last slide, the figures. Circulating hormones levels change, right? We know that insulin levels decrease and glucagon levels increase, and catecholamine levels also increase. And what happens to this hormones change, to the blood glucose levels, both insulin, glucagon levels increase will stimulate the glycogen, liver glycogen breakdown, right? And also stimulate gluconeogenesis. So during the exercise, because of the hormone levels change, particularly glucagon levels increase, and that will stimulate the hepatic or liver glycogen break down and release glucose and also makes more glucose through gluconeogenesis during exercise. So the liver makes glucose. So then released into circulation and used by skeletal muscle. And catecholamine levels also increase, which I already explained, right? The main purpose in our body to increase catecholamine is to stimulate lipolysis, right? And increase from this adipose tissue, right? Increase fatty acids released from adipose tissue. Another important thing is, remember, in the skeletal muscle, there's also triglycerides stored, right? So, and catecholamine increase can also stimulate HSL in skeletal muscle and break down triglyceride in the skeletal muscle, right? Yeah, question?
Yeah. If insulin is dropping so much, how does the glucose from gluconeogenesis and glycogen getting into cells?
Right, because glucagon levels is increased, right? So this will drive this pathway's activity. Break down glycogen and stimulate gluconeogenesis. And remember, what insulin does to glycogen is actually to stimulate glycogen synthesis and inhibits glycogen breakdown, remember, right?
So does the glucose stay in the cells and then it's used in the same cells? It never goes into the bloodstream?
Oh no, this happens in the liver,
Okay.
right? So then liver, you make more glucose from both breakdown glycogen and to make a new glucose from gluconeogenesis and released into the circulation and picked up by the muscle, right? And used in the muscle. So this sort of process. Yes?
Can I just ask you, is like, how does exercise stimulate the translocation of GLUT4 transporters so that you can still pick up the glucose without the insulin stimulus?
Oh, okay. That's a great question. So without insulin, remember we talked about the regulation of GLUT4 translocation from inside of cell to cell surface by two mechanisms, right? One is insulin, and the other one, that just by muscle fiber contraction itself, remember, can stimulate AMPK and then stimulate GLUT4 translocation, right? So the translocation of GLUT4 from inside of cell to the cell surface in skeletal muscle during the exercise also increase, but the mechanism is independent of insulin. Remember this, right? So, yeah, that's a great question. Thank you. Okay, so in addition to the hormone levels change during the exercise, the physiologically, right? And then the second thing is the blood flow to skeletal muscle is also increased. And what is the reason to increase the blood flow to the skeletal muscle? What is the purpose? It's
Increase oxygen?
yes, increase oxygen delivery to the muscle, right? And during exercise, you know the oxidation is increased in order to make ATPs to oxidize both glucose and fatty acids. So oxygen definitely the needed. I mean the needed for oxygen significant increase, right, during the exercise. So this will allow the more oxygen to be delivered to the skeletal muscle for oxidizing glucose and fatty acids for ATP production. And third thing is the muscle cytosolic calcium concentrations also increase. The muscle contractions requires calcium, right? So this is easy to understand. And the last thing is the AMP to ATP ratio is also increased. Why is it important? And what happens to like you just mentioned, you know, the GLUT4 translocation and fat oxidation? Yeah, AMPK, right? So AMPK can be stimulated by increased AMP to ATP ratio. Remember this, right? So AMPK is a kinase. It's very important in the regulation of energy metabolism. And two things that can be regulated by AMPK. Remember, we just talked about one, right? Group 4 translocation. So to the cell surface to increase glucose uptake into the muscle. So, and the second thing is, remember what AMPK does? Say that again.
Phosphorylates ACC.
Yes, that's pathway, right? And what is the downstream of ACC phosphorylation? Right, so increased beta oxidation. Remember that pathways, when AMPK is activated during exercise and increase the phosphorylation of ACC and leads to inactive of ACC, remember. And ACC is an important enzyme to catalyze the formation of marino-CoA, right? Remember that pathway. We're going to get that in a couple of slides later, all right? So two things that can be stimulated that is good for exercise. One is to increase glucose uptake because of increased group 4 translocation, right? And the second thing is the fatty acids oxidation. That's pathway that you already mentioned, right? To marino-CoA and CPT1 and transport activities and beta oxidation of fatty acids. So all these things together then increase the ability or capacity of skeletal muscle to oxidize the glucose and the fatty acids, right? And for ATP production. Yes?
So does insulin decrease because the muscle's losing glucose, or is it because that's what exercise does?
Yes, good question. So the reason why insulin levels decrease is because of we talk about glucose level decrease, right? And glucose level decrease primarily is because of muscle takes up a lot of glucose for oxidize for energy. So that's the main reason, right? So causing the decreased blood glucose level. So it's also the reason why exercise is good for diabetic patients, right? You exercise, you take a lot of blood glucose, so you are lowering, the blood glucose levels. Okay, now let's talk about the glucose utilization from rest to moderate and high intensity exercise. So initially, the exercise is initiated, then a lot of things change, right? The most important thing is that the glucose uptake into the muscle is increased, right? That leads to decreased glucose level and then stimulate the insulin, suppress the insulin release and stimulate glucagon levels. And once glucagon levels increase, and we try to make more glucose go to the circulation and used by the muscle. So one of the important sources of glucose is the liver glycogen, right? So glucagon increase and stimulate liver glycogen breakdown and release glucose. And it's also increase the glucose oxidation. For example, the dietary glucose, right? Is also being circulating in the circulation, and this can be uptake and oxidized in the muscle. And muscle glycogen store is also being the most important sources, right? The glucose during exercise. So muscle glycogen is different from the liver glycogen. The muscle glycogens break down to glucose 1-phosphate and fructose 1-phosphate and directly enter the TCA cycles for oxidation within the muscle cells, right? It's not going to release into circulation, right? Again, this is the pathway that we talked about already. The exercise increase group 4 translocation, increase glucose uptake, which is the mechanism is independent of insulin, right? This is AMPK mediated. As I mentioned earlier, that glycogen, muscle glycogen store is very important for energy of fuel utilization, particularly during the later stage from moderate to high intensity exercise. And also muscle glycogen store is significant influence affect the efficiency of exercise, right? It also affect the fat utilization. So in general, for example, if we have a less glycogen store, and particularly during the later stage when muscle glycogen is depleted, that will make the body, make the muscle utilize more fat, right, or rely on more fat utilization. That makes sense, right, during the later stage. And if we have higher muscle glycogen stores, that we less rely on the fat oxidation during the later stage or high intensity exercise, right? And remember, the fat utilization is actually decreased. The capacity is decreased during exercise. So that's why we need very important, we need more glycogen store in the muscle, so then you can have a less rely on fat oxidation during the high intensity exercise. So here I wanted to highlight the important important enzymes called pyruvate dehydrogenase complex. So pyruvate dehydrogenase complex activities basically reflects or represents the carbohydrate oxidation, right? If this pyruvate dehydrogenase activity is higher, that means you have more carbohydrate can be oxidized, right, being converted from pyruvate to acetyl-CoA and enter TC cycle for oxidation. That makes sense, right? So pyruvate dehydrogenase is very important, and this enzyme complex activities regulate by carbohydrate availability. So means that if we have more carbohydrate, for example, in skeletal muscle, if we have a more glycogen store, then pyruvate dehydrogenase activity is higher, increased. That will allow the more carbohydrate being oxidized, right? So the pyruvate dehydrogenase complex activity controls carbohydrate oxidation, right? So remember, this key enzyme is very important in the carbohydrate oxidation metabolism. So in the low muscle glycogen store, just want to go to the next slide. So if we have a less muscle glycogen store, right, and muscle glycogen depletion, that means we have less carbohydrate available, and under these conditions, the pyruvate dehydrogenase activity should be decreased, right? We don't have much carbohydrate being oxidized, so we need to shut down the pyruvate dehydrogenase, right, activities. So what happens to the following pathways if pyruvate dehydrogenase activities decrease, and then we have a less acetyl-CoA being generated from pyruvate, right? And this will have also leads to decreased production of marino-CoA by ACC. And remember, the marino-CoA also is a key substrate where we mentioned many times before, right? It's a key substrate that can inhibit the CPT1, which is a key transporter of fatty acids in the mitochondria, right? So decreased marino-CoA levels will lead to increased CPT1 activities. And this will allow the more fatty acid being transported from cytosol into mitochondria for beta oxidation, right? So this is sort of a mechanism, a pathway, how low carbohydrate store can lead to higher or more fatty acids oxidation. Right? Does that make sense? So that means you this pathway is need to be, stimulated if you don't have much carbohydrate stored in the muscle. And in the opposite condition, if we have enough or higher muscle glycogen stored, and then this pyruvate dehydrogenase will be high, right, will be stimulated, and will allow the glucose being oxidized further, right, to the acetyl-CoA. So acetyl-CoA levels will be increased. That will lead to have more malonyl-CoA being produced. And malonyl-CoA levels increase will suppress the CPT1 transporter activities, then will make less fatty acids being transported into mitochondria, right, to have a less fat oxidation. So this just kind of relationship, between the muscle glycogen store and the fat oxidation. So the reason why you have a lower muscle glycogen store and you have more rely on carbohydrate, more rely on fat oxidation, right, utilization. And here just wanted to clarify some concepts that we talked before and just make sure that you understand all this, and then we can go further. And first of all, the lactic acid production. So lactate usually is produced by partial oxidation glucose, right, through the glycolysis, and the reason particularly under the anaerobic conditions. So normally the lactic acid is produced during the anaerobic exercise, right, and by this type of muscle fibers, right. Type 2X, remember, is anaerobic fibers, usually have a lower oxidative capacity, higher glycolytic capacity, have, is a major sources of lactate production. And the second thing is about the exercise intensity. Remember, exercise intensity is determined, depend on the needs of ATPs. If the exercise requires more ATPs, that means this type of exercise is more higher intensity. But usually we define, measure the exercise intensity using oxygen consumption, remember. So the exercise intensity is normally related to the aerobic exercise, right? Because we measure the oxygen consumption to indicate the intensity of exercise. But of course, the main, the concept is actually is a need, depend on the need of ATPs. So anaerobic exercise, if they requires a lot of ATPs, is also considered high intensive exercise. But just because of a measurement, usually we measure oxygen consumption, so which mostly related to aerobic exercise. Does that make sense? I mean, just don't be confused about the definition and the way of we measure, the exercise intensity is a little bit different, right? And then the utilization of carbohydrate and fat. For anaerobic exercise, because does not require oxygen, right? The reason is it happened too quick. So oxygen is not enough time to be delivered to the muscle for oxidation. So basically, it's no oxygen and required, and is the and also glucose being oxidized through the anaerobic pathway, right? That just stop at the lactate. So for this type of exercise, anaerobic exercise, definitely the fat cannot be oxidized, right? So fat is not actually used for energy during anaerobic exercise, but only use the creating phosphate and carbohydrate within the skeletal muscle. right? It's very important energy source for anaerobic exercise. But aerobic exercise, since it requires energy in mitochondria, so fibers, the particular fiber type 1 and type 2A, so you'll be able to use both carbohydrate and fat as energy. All right. So how to increase the muscle glycogen store, since muscle glycogen store is so important, right, for the exercise efficiency. So it's a simple way, right? You just provide a glucose and to increase the glycogen synthesis in the skeletal muscle. Depend on the, but we're going to talk about more detail later. So usually you can give the, fructose containing drinks or the energy bar that contain a lot of carbohydrate. So you can, give them a exogenous source of glucose and allow the more carbohydrate glycogen being synthesized in the skeletal muscle. And why is increasing muscle glycogen stores so important for the race? Right, we already explained this. So here's the main reason, because of the ability of a muscle. I just want to emphasize again, right? So the ability of the skeletal muscle to utilize fat is reduced during the moderate to high intensity exercise, right? So during this time period, if you have a more glycogen store, there will be, that can be utilized for produce ATP, so allow you to maintain the similar levels of ATP. And the high glycogen muscle glycogen stores will also allow you to rely less on the fat utilization, right, during the moderate to high intensity exercise. So this is the main reason, just the physiological itself, because of the fat utilization is decreased during the later stage of exercise. So the muscle glycogen store is very important. All right, so here I wanted to explain some of the reasons that we talk why fat oxidation is increased from rest to moderate intense exercise, and why it's decreased during the moderate to high intensive exercise. So let's talk about the first time period, why fat oxidation is increased from low to moderate, or rest to moderate intense exercise. So the first reason is because of increased fatty acids availability. Remember, the hormone profiles change during the exercise, right? Particularly the catecholamine levels increase and insulin levels decrease because of the low level decrease of glucose levels. And these two hormone levels change will combine together, will stimulate the HSL activity in both adipose tissue, right, and increase the lipolysis. So adipose tissue will release fatty acids into circulation and pick up by muscle. So you have a more fatty acids available in circulation, and muscle will be able to pick up for use in the muscle. And also the blood flow is increased as well during the exercise, right? This will also allow to more fatty acids being able to deliver to the skeletal muscle, right? And remember, muscle itself also stores triglyceride. And similar mechanism, the catecholamine can also stimulate HSL inside of skeletal muscle, right, and to hydrolyze the triglyceride inside of skeletal muscle and break down, release fatty acids. So all this together, you can increase the fatty acids availability during this early stage of exercise. Any questions about this process? Okay, now the second reason why fat oxidation increase. And fat oxidations increase, and remember this pathway, right? This is very important. Again, slide. During exercise, because of AMP to ATP ratios increase, right? You realize a lot of energies and break down ATPs, right? Release energy. So a lot of ATPs being break down to AMP. So AMP to ATP ratio has increased, and this will stimulate the AMPK here, right? The AMPK is a kinase that catalyzes the phosphorylation of ACC, acetyl-CoA carboxylase, and this is key enzyme and leads to inactive, right? Once it gets phosphorylated and inactive, and this is a key enzyme for the formation of marino-CoA from acetyl-CoA, and so that will decrease marino-CoA level, right? And marino-CoA is the inhibitor of CPT1. So if marino-CoA levels decrease, then you will release inhibition of CPT1. So it will lead to increased CPT1 activities and allow more fatty acids being transported by CPT1 into mitochondria. So increase the beta oxidation, right? So this the two main mechanisms for reason why fatty acid oxidation is increased during resting to moderate intense exercise, right? One is availability of fatty acids increase, reasons because of hormone change, right? The catecholamine increase, insulin decrease, so increase in lipolysis from both adipose tissue and the skeletal muscle. So a lot of more fatty acids being released and available for oxidation in skeletal muscle. And second thing is because of this pathways, AMPK, activities increase, so a lot of more fatty acids being oxidized in the mitochondria. All right, so now the reason why fatty acid oxidations decrease from moderate and to high intensity exercise. One of the important reasons is because lower fatty acids availability in mitochondria, right? As the exercise continues and muscle oxidize a lot of carbohydrate and fatty acids, right? And this will lead to more acetyl-CoA molecules being accumulated, right? So, and the same mechanism, the acetyl-CoA can be converted to marino-CoA, right? And if marino-CoA levels increase, that will inhibit fatty acid transporter CPT1, right? So fatty acids transporter into the mitochondria, this process will be reduced. So try to remember these two things that causing the decrease the fatty acids available in mitochondria for oxidation. One is because of acetyl-CoA levels increase, right, due to oxidation of both glucose and fatty acids, and marino-CoA levels increase because this is converted from the acetyl-CoA, right? And this will inhibit CPT1 and decrease the transporter fatty acids into the mitochondria. Okay, now I'm going to give like two examples of aerobic versus anaerobic exercise, and we can hopefully we can understand better, how the energy fuels metabolized in two different types of exercise. So first, the aerobic exercise. A good example for aerobic exercise is a marathon. Right? Marathon usually runs like 26 miles, 3 hours. I don't know if it's a good performance. 3 hours, maybe between 2 and 3 hours, right? So an energy required for running a marathon actually about 2,868 kcal, right? And this, in order to get this much kcals, and we need to burn 750 grams of carbohydrate or 330 grams of fat. And remember that our body only has 400 to 900 grams of carbohydrate on board, right, in the fat condition, for example. And remember, we talked about this before, right? Actually, during the exercise, only half of muscle mass is involved in the marathon, in the exercise. So that means we only have a half, like 200 and 450 grams of carbohydrate available for oxidation. This definitely below the needs, right, is 750. So that means the muscle can not only just utilize carbohydrate for ATP production, will also need the other energy fuels, which is fat, right? So the muscle will need to utilize both carbohydrate and fat for ATP production during the marathon. So how can we know if muscle utilize different fuels? And this also related to the performance, actually, right? How much fat being used or how much carbohydrate being used by muscle. So how can we know this? So I'm going to introduce a concept. It's called the RQ. Remember, so estimation of a muscle fuel utilization. How we measure it, estimate. So RQ is a way to estimate the fuel mixture in the exercise, right? So how we measure the RQ? We usually measure the gas exchange in the lung. How much oxygen being taken up and how much CO2 being produced, and based on the ratio of the CO2 and oxygen, and then calculate the RQ, right? So how does this work? Because remember the glucose, amino acids, and fatty acids, they have a different chemical compositions, right? The oxygen, carbon, hydrogen, and amino acids has the nitrogen. So so if they oxidize and requires a different amount of oxygen, the different molecules, you cover glucose, fatty acids, or amino acids oxidize and requires different amount of oxygen, right? So based on this knowledge, then we can calculate the oxygen consumption versus the CO2 production. So RQ basically, this is the equation for RQ. RQ basically refers to the ratio of CO2 produced to oxygen consumed, right? The CO2 versus oxygen consumption. Let's take the example of a carbohydrate. So if 100% of energy is from carbohydrate, so if carbohydrate, 100% of carbohydrate is oxidized, and how much RQ will be. So this is the equation for the glucose oxidation, right? So glucose oxidized requires 6 molecules of oxygen and produce 6 molecules of CO2 and 6 molecules of water. So based on this equation, the RQ will be 6 molecules of CO2 produced and 6 molecules of oxygen used for complete oxidized glucose molecules, right? So RQ will be 1, right? For 100% of carbohydrate or glucose oxidation, RQ will be is 1. And what about the fat for fatty acids? Let's take a palmitate as the example. So palmitate is 16 carbons, right? It requires 23 molecules oxygen, and to produce 16 molecules CO2 and 16 molecules water. So again, if you calculate, the RQ will be around 0.7, right? So if complete 100% of fat is oxidized, RQ would be 0.7, and 100% carbohydrates oxidized, RQ would be 1. So what about RQ for oxidation of a mixture of carbohydrate and fatty acid? What do you expect RQ will be? Between, right? Yes, between 0.7 and 1. It depends, if you have a more carbohydrate used for energy and less fat, and the RQ will be more close to 1, right? And if you have more fat used and less carbohydrate used, then RQ will be more close to 0.7. So the mixture of carbohydrate and fat, RQ will be between 0.7 and 1. All right, any questions about the RQ? Yes?
So fatty acid oxidation is less efficient because of the chemical composition or the chemical molecule, or is it because of the metabolic pathway?
No, this is nothing related to the efficiency of the energy. Remember, we calculated before ATP production, right? One molecule of fatty acid is producing much more ATPs than compared to glucose metabolism. This is just a way to estimate how much fat being used for energy and how much fat the carbohydrate used for energy. So let's take, I have an example later. So for protein, because the structure is more complicated because I have an actual nitrogen, right? So its calculation is a little bit different, but RQ will be about 0.8. So here's an example, how we use this RQ to estimate the, how much fat or carbohydrate being used during the exercise. For example, the RQ for a marathon runner, so RQ for skeletal muscle, in like recreation marathon runners, for example, in the first 30 minutes we're running, right? When we measure the RQ of these individuals, then the RQ is 0.9, between 0.9 and 1, right? And after 2 hours running, and when we measure the RQ again, and the RQ actually decreased to 0.8 to 0.85. So what does this information tell us about the fuel utilization by this individual, by this runner?
You burn more fatty acids after 2 hours.
Yes, thank you, very, very good. So in the, after, in the 30 minutes, during the first early 30 minutes running, right, they use more carbohydrate, right? This runner used more carbohydrate for ATP production in his or her muscle, right? And after 2 hours running, see the utilization of the fuel actually switched a little bit, right? They started to utilize more fat for energy and less carbohydrate for energy as compared to initial 30 minutes time period, right? So just understand how RQ is calculated and what does RQ values mean, right? So help you to evaluate or estimate, the fuel utilization in skeletal muscle during exercise. I think we have about 5 minutes. So now I'm going to talk about the chain unchain. Remember we mentioned earlier after chaining that muscle can change, can adapt to utilize the different energy source, right? Different energy fuels And also you can increase the maximum oxygen consumption. Remember, the oxidative capacity can be also increased after training. So here, just to make a comparison between the recreational versus the elite runners, and what is the difference between these two different type of runners in the fuel utilization. So let's see the recreational runners. During the exercise period, the running efficiency actually gradually decreased. And they have a more significant switch of fuel utilization from carbohydrate in the beginning and to the fat in the later stage of exercise, of race, right? So the recreation runner means that at the later stage, they more rely on the fat oxidation. And because the fat oxidation is reduced, right, so that will significantly affect the running efficiency for the recreation runner. So you have a more significant switch from carbohydrate at the beginning and to the fat in the later stage of exercise, right? And second thing is that they have a higher lactate production because of they recruit more type 2X fibers during the exercise, right? So in other words, the recreation runner, their skeletal muscle have a more percentage of type 2X, right? So that's why they make more lactate and less or lower oxidative capacity. And compared to elite runners, and their switch or the efficiency reduction is quite small, right? They can pretty much efficiently, run exercise. And the utilization, the switch from carbohydrate to fat, the switch is much small, right? They're pretty constant to utilize the different sources of energy, carbohydrate and fat. So they have a less lactate production because they have more type 1 or type 2 fibers and less type 2X fibers, right? So that's why they have a higher oxidative capacity. And they can use the fat as the energy, right? Because of high capacity, oxidative capacity. Yes?
So on the slide before this, where it says capacity for fat utilization is small, is that like within the first hour? Or is it talking about, and then when it switches to more fat and less carbs after like a long time?
Right, you mean the recreation runners?
Yeah.
Yeah, because the first stage, the early stage they use more carbohydrate, and the later stage they tend to try to use more fat. But because of fat, the oxidation capacity is lower, decreased, right? So they actually weren't able to efficiently utilize fat to produce ATPs, right? All right, any other questions? Okay, so let's take a 10 minutes break, and then we have quiz 6, right? And also, I want to remind you guys, if you have done the evaluation for my teaching, please do it, and I really appreciate